Okay, let's dive in. We spend so much time thinking about, the cells in our bodies, like all 37 trillion of them.
The little workers.
Right, exactly.
Mm.
Neurons firing, muscle cells contracting, all that stuff. But imagine, just for a second, you could somehow dissolve away every single one of those cells. What would actually be left?
Ah, that's a fascinating thought experiment. You'd be left with this almost ghostly replica of your entire body.
Ghostly.
Yeah, a precise architectural model, but made entirely of protein networks. It would look just like you, but without any cells inside.
Wow. And that structure. That's the extracellular matrix, the ECM.
That's it precisely, the ECM.
Because, for the longest time, wasn't the ECM just seen as, well, background noise? Like biological packing peanuts.
That's a good way to put it. Yeah, it was often dismissed as just scaffolding or even just glue, the stuff holding cells together.
But the sources we looked at paint a completely different picture. There's this whole universe of activity outside the cell. It's way more than just glue.
And that's the absolutely critical shift in perspective we need to make. The ECM is anything but passive. It's dynamic.
Dynamic how?
It's constantly being remodeled. It's this huge protein-based communication network. And it really dictates how cells behave, whether they survive, what they become.
So it's telling the cells what to do.
In many ways, yes. And it makes up a significant chunk of almost every single tissue in your body. It's not a minor component.
Okay, so for anyone listening, especially students or anyone curious about biology, understanding this matrix is like a shortcut, right?
It really is. It helped you understand almost every process, physiological or pathological.
Like what specifically?
Well, think about how we heal, how organized tissue regeneration actually works, or why certain diseases progress the way they do, things like chronic fibrosis in the liver or lungs.
The scarring diseases.
Exactly, and even genetic disorders. Why do they manifest the way they do? Take Marfan syndrome, that's fundamentally a problem with ECM structure, leading to those devastating consequences.
So our mission today is really to get past that simple scaffolding idea.
Right. We need to see the ECM as this active functional entity. Yes, it provides mechanical support, it anchors cells, guides them when they migrate, helps them maintain their polarity, their proper orientation.
Okay, the physical stuff.
But the fascinating part is that it's also a powerful signaling hub, and even a kind of emergency reservoir.
A signaling hub. That sounds like constant chatter, like a two-way conversation between the cells and everything around them.
Absolutely. That's a great way to think about it. The ECM directly regulates cell proliferation because it binds onto essential growth factors. It can hold them, display them, keep them inactive until they're needed, or make them active.
So it controls the signals?
And it signals directly into the cell itself through specialized receptors. It tells the cell, what to do, where to go, based on the environment.
And you mentioned structural integrity. This is literally the foundation for healing, for life itself.
It absolutely is. Look, if you destroy the organized structure of the ECM, especially key parts like the basement membrane or the underlying stromal scaffold, then you simply cannot get organized tissue regeneration. The body can't rebuild properly. It defaults to disorganized repair.
Which we see as scarring, fibrosis.
Precisely. Scarring is a failure of organized regeneration because the blueprint, the ECM structure, was too damaged. And beyond that, the ECM creates critical microenvironments. Think about the kidney.
Filter.
Exactly. The basement membrane there isn't just a boundary. It's a vital part of that complex, highly selective filtration system. It lets some things through, holds others back. It's incredibly specialized.
Okay, this is clearly much more complex than just glue.
Yeah.
Let's get into the architecture then. You said it's not uniform.
Not at all. It has two main forms, structurally and functionally distinct. We need to differentiate them clearly.
So what are they?
You've got the interstitial matrix, or IM, and the basement membrane, or BM. Think of the IM as the general filler, the cushion, and the BM as a highly specialized, organized boundary layer.
Like comparing, I don't know, the insulation foam in walls versus the specialized vapor barrier layer.
That's not a bad analogy, actually.
Yeah.
Yes, let's start with the IM, the body's cushion. Where do we find this stuff?
Okay, the cushion, where is it?
The interstitial matrix is, well, in the interstices. It occupies the spaces between cells and connective tissue, the stromal cells. And it also fills the gaps between the main functional cells of an organ, the parenchyma, and their underlying support structures, like blood vessels or smooth muscle layers.
So it's really widespread. And who makes it?
It's primarily synthesized by mesenchymal cells. The main players are the fibroblasts. They're the key architects of the IM.
And architecturally, you described before as amorphous, a semi-fluid gel. What does that actually mean for its function?
Amorphous just means it doesn't have a highly ordered repeating structure like a crystal. It's more like a complex three-dimensional mesh. And being a semi-fluid gel is key to its cushioning function.
Cushioning against what?
Against compression. Think about the constant mechanical stresses your body deals with. Your gut contracting during peristalsis, your bladder filling up, your arteries pulsing with every heartbeat.
Right, constant movement and pressure changes.
Exactly. Those actions compress and strain the tissues. The fluid trapped within the IM's protein meshwork acts like a hydraulic shock absorber.
So the water component is crucial.
Absolutely. It resists the compression, preventing rapid squashing from tearing cell connections or collapsing vital tubes. It's a passive but absolutely essential protective layer. It allows tissues to deform and then return to shape.
That's something we totally take for granted, isn't it? The ability to move without constantly ripping things internally. Okay, so what's this cushion actually made of? What are the building blocks?
The main solid components of the IM are things we'll get into more detail on, but basically, various types of collagens, both fibrillar and non-fibrillar, fibronectin, elastin for stretchiness, those superhydrated proteoglycans, and hyaluronate.
Okay, a whole mix of proteins and sugars. Now let's contrast that gel-like cushion with the other form, the basement membrane, BM. Yeah. You call this the organized boundary.
Right. The basement membrane isn't just filler, it's a highly organized layer of interstitial matrix components, but arranged into a specific sheet-like structure.
Where do we find this sheet?
It forms a specialized surface directly underlying and supporting epithelial cells, the cell's lining surfaces and cavities. It's also found around endothelial cells, which line blood vessels, and around smooth muscle cells. It provides a platform they recognize and attach to.
And who builds this specialized layer? Is it still the fibroblasts?
Here's a key difference. The synthesis of the BM is actually a joint effort. Yes, unlike the IM, which is mostly made by mesenchymal cells like fibroblasts, the components of the BM are synthesized by both the overlying epithelial or endothelial cells and the underlying mesenchymal cells.
So it's like a shared construction project at the boundary. That makes sense. And structurally, it's not a gel.
No, architecturally, it leaves the 3D gel behind and forms a flat, sort of lamellar mesh. Think of it like a very fine, interwoven mat.
But you also said it's porous, even though it's called a membrane. That seems counterintuitive.
It does, but membrane here refers more to its sheet-like form acting as a boundary, not necessarily an impermeable barrier like a cell membrane. It's definitely porous.
Okay. And what are the main components giving it this specific structure?
The key players are different here. It's mainly specialized non-fibrillar type 5e collagen and the adhesive glycoprotein laminin.
And its main job?
It provides that crucial structural support and boundary function we mentioned. And in places like the kidney glomerulus, its specific composition and porosity are absolutely critical for filtration, acting as that selective sieve, letting water and small solutes pass while holding back large proteins in cells.
Okay, two distinct environments. The general cushion, IM, and the specific boundary, BM. Now let's dig into the actual materials, the structural backbone. If it's a building, these are the steel beams and maybe the rubber bands.
That's a great analogy. Let's start with the steel. Collagens. They are all about tensile strength.
Tensile strength. Resisting pulling forces.
Exactly. Collagen is incredibly good at resisting stretching and tearing. It's actually the single most abundant protein in your entire body.
Wow. And structurally, what is it?
The basic unit is a molecule made of 3 polypeptide chains braided together very tightly. It forms this characteristic rope-like triple helix structure. And there isn't just one type we know of, about 30 different types of collagen, each adapted for its specific job and location.
30 types, okay. But when people think collagen, they usually think of the really tough stuff, right? Like in tendons or bones.
Yes, those are the fibrillar collagens, like types 1, 2, 3, and V. These are the ones that bundle together.
How do they go from that tiny triple helix rope to these massive strong fibers we see in tissues?
Well, the individual triple helices first align and aggregate into larger structures called fibrils. Initially, they're held together by hydrogen bonds within the helix itself. These fibrils are the main components of bone, which is mostly type of collagen, but also tendons, cartilage, skin, and importantly, scar tissue that forms during healing.
But hydrogen bonds alone don't seem strong enough for, say, an Achilles tendon.
Yeah.
What gives it that incredible strength?
That's the crucial next step. The real strength, the ability to withstand huge forces, comes after the fibrils assemble. It comes from lateral cross-linking.
Cross-linking, like welding the ropes together.
Exactly like that. Covalent bonds form between adjacent triple helices within the fibril. This cross-linking process relies on a specific chemical modification, the hydroxylation of certain lysine residues on the collagen chains.
Lysine hydroxylation.
Okay, this modification allows those strong covalent bonds to form, effectively welding the individual ropes into an integrated, incredibly strong cable. That's where the real tensile strength comes from.
And this chemical step, the hydroxylation, this is where diet comes in, right? The classic vitamin C connection.
Precisely. This is a perfect illustration of structure meets nutrition, the enzyme that performs that absolutely critical lysine hydroxylation step.
The one needed for the cross-links.
Yes, that enzyme, lysyl hydroxylase, absolutely requires vitamin C ascorbic acid as a cofactor to function. Without vitamin C, it doesn't work.
So if you don't have enough vitamin C, your body can still make the collagen chains. It can braid the helix.
It can. It can build the basic ropes, but it cannot perform the hydroxylation step properly.
Meaning no cross-links, no welding.
Exactly. The collagen heliases assemble, but they can't be covalently linked together. The resulting structure is weak, unstable, and easily broken down.
And that's scurvy.
That is the molecular basis of scurvy. It explains everything. Why children with severe deficiency get skeletal deformities, their bones lack properly strengthened collagen. Why adults heal terribly, why their blood vessels become fragile and they bruise easily or bleed spontaneously. It's not a failure to make collagen, it's a failure to finish it, to give it architectural integrity.
Fascinating. Okay, so those are the main fibrillar types. You also mentioned non-fibrillar collagens doing different jobs.
Yes. These don't form those big, tough fibrils. Type 4 collagen, for instance, is the one that forms the flat, mesh-like network structure of the basement membrane we just talked about.
The specialized boundary layer.
Right. Then there's type 7 collagen. Its job is to form anchoring fibrils. These are like tiny staples that physically connect the epidermis, the outer layer of skin, to the underlying dermis.
So if that fails...
Mutations in type 7 collagen cause certain inherited blistering skin diseases because the skin layers literally can't stay attached under minor stress. And then you have others like the fessic collagens, type X is an example, which help regulate the diameter of the larger fibrillar collagens, making sure they assemble correctly.
And just like with the vitamin C issue, if there are genetic flaws in the collagen proteins themselves or in the enzymes that process them, the consequences are systemic and severe.
Absolutely. We see this clearly in diseases like osteogenesis imperfecta or brittle bone disease, often caused by defects in the structure of type I collagen itself. The bones just aren't strong enough.
And Ehlers-Danlos.
Yes, the Ehlers-Danlos syndromes, EDS, are a whole group of disorders affecting connective tissue. They can be caused by mutations in various collagen genes, or importantly, in enzymes involved in collagen processing, like that same lysyl hydroxylase needed for cross-linking. Patients often have hypermobile joints, stretchy skin, fragile tissues. It's all down to weakened connective tissue structure.
Okay, we've covered the steel beams, the strengths from collagen. Now, what about the bounce back? The rubber bands, that's elastin, right?
That's elastin. Its job is all about recoil. It gives tissues the ability to stretch under stress and then snap back to their original shape when the stress is removed. Elasticity.
Preventing permanent deformation. Where is this most critical?
Think anywhere that needs to expand and contract repeatedly. The large arteries, like the aorta, are prime examples. They have to balloon out slightly with every heartbeat and then snap back to push the blood forward.
Right, absorbing the pulse wave.
Exactly. Also crucial in cardiac valves, which open and close constantly. The lungs need it to expand and recoil with breathing. The skin needs it to return to shape after being pinched or stretched. Even ligaments and the uterus rely heavily on elastin.
So structurally, how does it achieve this stretchiness? It can't be like the rigid collagen ropes.
No, it's very different. Elastin fibers have a core made of the elastin protein itself. This core is surrounded by a mesh-like network made of another protein called fibrillin.
Fibrillin.
Yeah.
Okay.
The elastin protein has large segments that are hydrophobic, meaning they don't like water. When the tissue is relaxed, these segments tend to clump together. When you stretch the tissue, you pull these segments apart, exposing them that stores potential energy, like stretching a rubber band.
And what holds it together so it doesn't just break?
Elastin has unique cross-links different from collagens called desmosine and isosmosine cross-links. These hold the elastin molecules together. When the stretching force is released, the hydrophobic segments snap back together, recoiling the whole tissue.
And that mesh around it, the fibrillin, you said that leads us to another major condition.
Yes, the fibrillin scaffolding is critical. Defects in fibrillin synthesis cause Marfan syndrome.
Ah, right. The condition associated with being very tall, long limbs, and serious heart problems.
Exactly. The skeletal abnormalities are one part, but the really dangerous aspect is the weakened, dilated aortic walls. Because the aorta relies so heavily on properly formed elastic fibers, which need that fibrillin scaffold defects lead to aneurysms and potential rupture.
But wait, if fibrillin is just the scaffold for elastin, why does messing it up cause such widespread problems, even beyond elasticity? How does it affect bone growth, for example?
Because fibrillin is doing more than just being a scaffold. It's another example of the ECM controlling signals. Fibrillin plays an absolutely key role in regulating the activity of transforming growth factor beta, TGF.
TGF-beta again. We talked about that with scarring.
Yes, it's a major player. Normally, much of the TGF-beta secreted into the ECM is kept inactive, latent, and fibrillin is what sequesters it. It physically binds to latent TGF-A and keeps it tucked away.
So the fibrillin mesh isn't just holding elastin, it's like a cage for TGF-beta.
That's a perfect way to think about it, a cage. Now, in Marfan syndrome, the fibrillin is defective. It can't properly build the elastic fiber scaffold, and it can't effectively cage the latent TGF-beta.
Meaning too much active TGF-beta gets loose.
Precisely. You get excessive, unregulated TGF-off signaling. And this contributes significantly to the problems seen in Marfan's, including the abnormal bone growth and, critically, the inflammation and breakdown in the aortic wall. It's a structural defect causing a signaling disaster.
Wow, okay, so structure links directly to signaling again. We've got strength, collagen, resilience, elastin. What about the cushion aspect we started with, the hydrated part?
Right. Now we move from the fibrous proteins to the stuff that fills the space and gives tissues their resistance to compression. This is the domain of proteoglycans and hyaluronan.
Resisting squashing forces. And lubrication, too, you mentioned.
Yes, absolutely critical for lubrication, especially in joints. That smooth gliding motion in your knee, thank proteoglycans in the cartilage. Their main function, though, is forming these highly hydrated gel-like substances that resist compressive forces.
Okay, let's unpack the structure. It sounds complicated. You mentioned a bottle brush analogy earlier.
It's a great visual. The structure is hierarchical. It starts with a very long central filament made of hyaluronan, also known as hyaluronic acid. It's just a long chain of repeating sugar units.
Okay, the backbone.
Attached to this hyaluronan backbone, like bristles sticking out from the handle of a round brush, are numerous proteoglycan molecules.
And what's a proteoglycan?
A proteoglycan itself has a core protein, and attached to that core protein are many long chains of specialized polysaccharides called glycosaminoglycans, or GAGs.
GAGs, like chondroitin sulfate, things you hear about for joint health.
Exactly. Chondroitin sulfate, keratin sulfate, heparin sulfate, those are all types of GAGs. So you have GAGs attached to a core protein, making a proteoglycan. And then many proteoglycans attached to a central hyaluronan chain. It forms these huge, complex aggregates.
A massive molecular brush indeed. What makes this structure trap so much water and create that gel?
It all comes down to electrical charge. The JAG chains, especially the sulfated ones like chondroitin sulfate and heparin sulfate, are packed full of negatively charged groups, just loaded with negative charge.
Okay. This dense negative charge strongly attracts positively charged ions, catations, from the surrounding fluid, mostly sodium ions. Na+.
Opposites attract.
Right. And wherever you get a high concentration of sodium ions, water follows passively by osmosis. Lots of water rushes in trying to balance the concentration.
So the negative charges on the JAGs pull in sodium, and the sodium pulls in tons of water.
Precisely. And this creates that viscous, swollen, gel-like matrix. It's pressurized by the trapped water. That's what gives tissues their turgor, their ability to resist being squashed. You are essentially walking around on microscopic self-inflating water cushions.
That's incredible. So that's the primary mechanical role. But you hinted earlier these also link back to signaling, the growth factor reservoirs idea.
Yes, this is a crucial secondary function. Those GAG chains, particularly heparin sulfate found on many proteoglycans, act as binding sites for numerous growth factors.
Which ones? Things like fibroblast growth factor, FGF, hepatocyte growth factor, HGF, vascular endothelial growth factor, VEGF. Many important signaling molecules get secreted into the ECM and immediately bind to the heparin sulfate chains.
So the matrix acts like a storage depot, a library holding these factors.
Exactly. It keeps them localized right there in the tissue, but often in an inactive or latent state, bound to the proteoglycan. They're held in reserve until they're needed.
Needed when? like during injury.
Precisely. When tissue is damaged or needs to remodel, specific enzymes get released that can actually clip the growth factors off the proteoglycans or modify the proteoglycan itself, releasing the factor in an active form right where it's needed for repair or angiogenesis.
So the matrix controls the timing and location of growth factor activity.
Yep.
And what about molecules that actually bridge the matrix right to the cell surface?
Good question. That brings us to things like the cell surface proteoglycans. A key example is syndekin. Yes. Syndekin is actually an integral membrane protein, meaning part of it crosses the cell membrane and sits inside the cell, while another part sticks out into the ECM.
Okay, spanning the boundary.
Right. The part sticking out has JAG chains, usually heparin sulfate, just like the big matrix proteoglycans, so it can bind and concentrate growth factors like BFGF right at the cell surface.
Bringing the signal right to the cell's doorstep.
Exactly. But the critical part is the piece inside the cell, the cytoplasmic tail. This tail interacts directly with the cell's internal scaffolding, the actin cytoskeleton.
Connecting the outside world to the cell's inner structure.
Precisely. This makes syndekin a transducer. It allows the binding of factors in the ECM, or even just the physical properties of the ECM it's embedded in, to directly influence the cell's internal machinery, affecting things like cell shape, movement, migration, and proliferation. It's a direct communication line.
Okay, this is getting intricate. We've got the structure, the cushion, the reservoirs. Now, what about the molecules that handle the direct connections, the communication specialists?
Now we're talking about the molecules that really organize the matrix and mediate the cell's interaction with it. These are the adhesive glycoproteins and their receptors. They manage cell-cell interactions, cell ECM interactions, even ECM interactions. They're crucial for attachment, movement, and organization.
Let's start with the glycoproteins themselves. The big names seem to be fibronectin and laminin. What does fibronectin do?
Fibronectin is like the master organizer, the multi-tool of the interstitial ECM. You find it pretty much everywhere in connective tissue. It's made by various cells, fibroblasts, immune cells like monocytes, endothelial cells lining blood vessels. It exists in different forms too, some woven into the matrix, some soluble in blood plasma.
What makes it a master organizer? What can it bind to?
Its structure is key. It has distinct domains like different tools on a Swiss Army knife. Some domains bind specifically to other ECM components like collagen, fibrin, which forms blood clots, and heparin, found on proteoglycans.
So it links different parts of the matrix together.
Yes. And crucially, it also has domains that bind directly to cell surface receptors, the integrins, which we'll get to. This allows it to physically link cells to the surrounding matrix.
And you mentioned its role in healing earlier, like an emergency responder.
Absolutely. When you get wounded, fibronectin from your plasma quickly gets incorporated into the initial blood clot, forming a temporary matrix. This fibronectin mesh then acts as a scaffold.
A scaffold for what?
For the next stages of healing. Cells like fibroblasts crawl along it to deposit new permanent ECM. New blood vessels grow along it, angiogenesis. Skin cells migrate over it to close the wound, reepithelialization. It provides the temporary roadmap for organized repair.
Okay, so fibronectin is the versatile workhorse of the general matrix. How does laminin differ? You said it was more location-specific.
Exactly. Laminin is the star player almost exclusively found in basement membranes. It's the most abundant glycoprotein in that specialized layer under epithelia and endothelia.
And its structure?
It's a large protein, usually shaped like a cross, made of 3 different chains, a heterotrimer. Like fibronectin, it has different domains. Its main job is to connect the cells sitting on the basement membrane via their integrin receptors to the core components of the BM itself, primarily type 5 collagen and heparin sulfate proteoglycans. It anchors the cells to their foundation.
So mostly structural anchorage.
That's a key role, but again, it's not just passive anchoring. Laminin is also a potent signaling molecule. Different types or isoforms of laminin can strongly influence cell behavior. They modulate proliferation, differentiation, and motility.
How so?
For example, specific laminin variants in the developing nervous system help guide growing axons to their correct targets. In epithelia, the type of laminin can help determine which side of the cell is the top, apical, and which is the bottom, basal, establishing cell polarity. So it provides structural context and instructional cues.
Okay, both fibronectin and laminin connect to cells via receptors. Let's get to those receptors now, the integrins, the handshake molecules.
The handshake, the translators, the crucial link. Integrins are absolutely central. They're a huge family of proteins that sit across the cell membrane, transmembrane proteins.
Family, meaning there are lots of different types.
Lots. They're heterodimers, meaning each integrin receptor is made of two distinct protein subunits, an alpha subunit and a beta subunit. And there are many different alpha and beta subunits that can pair up in various combinations, creating a wide variety of integrins, each specialized to bind specific ECM molecules or even molecules on other cells.
And how do they actually grab onto things like fibronectin or laminin? You mentioned a tiny code earlier, the RGD motif.
Yes. This is one of the most elegant findings in cell biology. Many key ECM adhesion proteins, including fibronectin, laminin, another one called vitronectin, even fibrinogen involved in clotting, contain a specific tiny sequence of just 3 amino acids. Arginine, glycine, aspartic acid.
RGD.
RGD.
3 letters of the protein sequence.
Just 3 letters. And the integrins are specifically designed to recognize and bind tightly to this RGD motif on their target ECM proteins. It's like a universal docking sequence, the common language used for cell matrix adhesion across many tissues.
That's amazing. Such massive structures held together by this tiny molecular code. So the integrin is the hand reaching out from the cell, grabbing the RGD on the matrix. Where does that hand connect inside the cell? What happens next?
It connects to the cell's engine room and control panel. Integrins aren't just anchors. They are true signal transducers. When an integrin binds to its ECM ligand, like fibronectin, via the RGD motif, it triggers events inside the cell. The integrins cluster together on the membrane, and they recruit a whole bunch of other intracellular proteins to form a complex structure called a focal adhesion.
Focal adhesion, okay.
This focal adhesion complex acts as both a physical link and an information hub. Physically, it connects the ECM outside directly to the cell's internal cytoskeleton, the network of actin filaments that gives the said shape and allows it to move.
So outside connects straight to inside structure.
Directly, but it's also informational. The act of binding, the clustering of integrins, activates signaling pathways inside the cell. Enzymes like tyrosine kinases get switched on, triggering cascades that control critical cell behaviors.
Like what?
Things like cell locomotion, how the cell crawls, cell proliferation, whether it divides, cell shape, how it organizes itself, even differentiation, what type of cell it becomes. The signals coming from the ECM through the integrins are fundamental inputs for all these decisions.
So the cell is literally feeling its environment through the integrins, like how stiff or soft the matrix is.
Exactly. The mechanical properties of the matrix, its stiffness, which depends on things like collagen cross-linking, are sensed by the integrins and translated into biochemical signals inside. A cell on a stiff matrix behaves very differently from one on a soft matrix.
And clinically, this is important for immune cells.
Hugely important. Think about inflammation. White blood cells, leukocytes, need to get out of the bloodstream and into the infected tissue. How do they do it?
They stick to the blood vessel wall and squeeze through.
Right. That sticking process, the firm adhesion to the endothelial cells lining the blood vessel, and their subsequent migration across that barrier that's heavily dependent on integrins being activated on the leukocyte surface, allowing them to grab onto molecules on the endothelium. It's integrin-mediated adhesion and migration. They also play a key role in getting platelets to clump together to form a blood clot.
Okay, so the ECM isn't static. It talks to cells via integrins, which implies the ECM itself must change, right? It gets remodeled.
Absolutely. This is a vital concept. The ECM is not like concrete poured once and left forever. It is constantly, dynamically being synthesized and, just as importantly, degraded or broken down.
Why? When does this happen?
This remodeling is fundamental. It happens during embryonic development as tissues take shape. It happens constantly at a low level for normal tissue maintenance. It ramps up significantly during tissue repair after injury. But it also happens pathologically in chronic fibrosis, where there's too much deposition. And critically, it's essential for tumor invasion, and metastasis cancer cells have to remodel the ECM to escape.
So this balance of building up and breaking down is key. What controls it? We keep coming back to TGF-β. Is the master switch here too?
TGF-β is arguably the single most important factor controlling ECM deposition and remodeling, especially in the context of repair and fibrosis. But it's famously complex.
Complex how?
It's highly pleiotropic. That means it can have many different, sometimes even opposing effects, depending on the context.
Like what?
Depending on the specific cell type, the other signals present, the receptors involved. TGF-β can inhibit cell growth or stimulate it. It can promote differentiation. But its most prominent role, the one we usually focus on conceptually, is driving scar formation.
Okay, let's focus on that pro-fibrotic, pro-scarring role. How does it actually build up the matrix and make it stick around?
It tackles both sides of the equation, synthesis and degradation. First, production. TGFF strongly stimulates mesenchymal cells like fibroblasts to produce more of the major ECM components, more collagen, more fibronectin, more proteoglycans. It tells them to build.
Okay, step 1, make more stuff.
Step 2, prevent breakdown. TGF-β actively inhibits the degradation of the ECM, especially collagen. It does this in two main ways.
How?
It decreases the activity and production of enzymes called matrix metalloproteinases, or MMPs. These MMPs are the primary tools cells use to chew up and break down ECM components. So TGF-β dials down the chewing enzymes.
Makes sense. And the second way?
Simultaneously, it increases the activity of molecules called tissue inhibitors of proteinases, or TIMPs. As the name suggests, TIMPs bind to MMPs and neutralize them. So TGF-β reduces the degraders, MMPs, and increases their inhibitors.
A double whammy to stop breakdown.
Yeah.
Build more, break down less. That leads to accumulation.
Exactly. It strongly shifts the balance towards matrix accumulation, which is great if you need to quickly form a scar to patch a wound.
But bad if it doesn't switch off, like in chronic diseases.
Precisely the problem. In chronic inflammation, maybe from a persistent virus in the liver, high blood pressure damaging the kidney, or constant irritation in the lungs, the signals that trigger TGF release don't go away.
So it just keeps telling the cells to build scar tissue.
Continuously, you get this relentless, excessive deposition of disorganized matrix, primarily collagen. This is fibrosis. The organ gets progressively stiffer, normal tissue architecture is destroyed, and eventually function fails. It's TGF-A signaling run amok.
Okay. And related to remodeling is it other idea that ECM is a growth factor storage depot, the timing mechanism.
Yes, let's revisit that. The ECM isn't just structure. It's a reservoir for these latent inactive factors, held captive by components like the heparan sulfate on proteoglycans. We mentioned FGFs earlier.
Right. So if the growth factor is locked up, how does the body specifically release it only when and where needed? Say, at a wound site.
The release mechanism is often tied directly to the matrix breakdown itself. It involves proteolysis enzymes cutting proteins.
Like the MMPs.
Yes. MMPs or other proteases, like serine proteases, are often released or activated at sites of injury or inflammation. These enzymes start chopping up the ECM components. As they degrade the proteoglycans, for instance, they can physically cleave the growth factor, like BFGF, away from its heparan sulfate binding site.
Ah, so the act of dismantling the old scaffold literally unlocks the signals needed to build the new one.
Exactly. The factor is released from its cage, becomes biologically active, and can then stimulate nearby cells to proliferate, migrate, and start the repair process. It's an incredibly elegant way to couple matrix degradation with the initiation of regeneration, ensuring the signals are delivered precisely at the right time and place.
So the cell's getting multiple inputs simultaneously.
It really is. It's sensing the physical environment via integrins. How stiff is it around here? And it's sensing the biochemical environment via receptors for growth factors that have just been released. Okay, time to divide. The cell integrates all these signals to produce a coordinated response. Move here, divide now, become this type of cell.
This really shifts the whole perspective. Okay, so knowing all this complexity, how is this changing how we approach diseases where the ECM is involved? This takes us into the newer advancements, right?
Absolutely. This deeper understanding is opening up really fascinating avenues for medical intervention. We're moving beyond just targeting the diseased cells and thinking about how to treat the diseased environment, the matrix itself.
Let's start with targeting fibrosis. Since TGFE is the bad guy in chronic scarring, are we just trying to block it?
Well, that was the initial thought, and there's still research there. But the problem is TGFE does so many other important things in the body. It's involved in immune regulation, cell growth control.
So blocking it everywhere could cause major side effects.
Exactly. It's too pleiotropic. So a lot of current, more advanced research is focusing further downstream, trying to identify and inhibit specific signaling pathways activated by TGFO that are uniquely important for the pathological scarring process.
Like what?
Like finding ways to specifically modulate that MMP-TIMP balance only in the fibrotic tissue. Can we selectively boost MMP activity just enough to break down excess collagen, or selectively block the TIMPs that are preventing breakdown without causing chaos elsewhere? The goal is more targeted modulation, trying to nudge the balance back towards controlled remodeling instead of relentless deposition. Think conditions like idiopathic pulmonary fibrosis or liver cirrhosis. That's where this is desperately needed.
Okay, targeted breakdown. What about the ECM's role in cancer? That seems like another huge area.
It's absolutely central, especially in tumor progression and metastasis. We now understand the tumor microenvironment, largely shaped by the ECM, is critical.
How so? We talked about stiffness.
Yeah.
Right. The physical properties matter immensely. Tumor cells often thrive in and are promoted by a stiffer matrix. Techniques like atomic force microscopy can actually measure the stiffness of tissues around tumors. And often, increased stiffness, usually due to excessive deposition and cross-linking of type I collagen, correlates strongly with more aggressive tumors and worse outcomes for the patient.
So the stiff matrix helps the cancer. And how does it help cancer spread, the metastasis part?
It provides the highway. For a tumor cell to metastasize, it first has to break through its local barriers, like the basement membrane. That requires activating those MMPs we talked about, the matrix-degrading enzymes.
It escapes its local area. Then what?
Then it needs to migrate through the interstitial matrix to reach blood vessels or lymphatics. That matrix, often guided by aligned fibers of fibronectin or collagen, becomes the physical track or road the cancer cell crawls along.
So therapies could target this road.
Exactly. Advanced strategies are looking at ways to disrupt this process. Can we target the specific integrins that cancer cells use to grip the matrix fibers and pull themselves along? Can we inhibit the enzymes, like lysyl oxidase, that perform the collagen cross-linking, making the matrix less stiff and harder for tumor cells to navigate? The aim is to disrupt the tumor microenvironment and physically block the escape routes.
Fascinating. Roadblocks for cancer cells. And the third big area you mentioned, regenerative medicine, using the ECM as the scaffold.
This is maybe the most exciting frontier, directly building on that concept of the ECM as the scaffolding for tissue renewal. We're moving way beyond just growing cells on flat plastic dishes.
What are researchers doing instead?
They're engineering sophisticated three-dimensional scaffolds that are designed to mimic the natural ECM of the tissue they want to regenerate, whether it's heart muscle, liver tissue, cartilage, bone.
Mimic how? Just the structure.
Structure is part of it, getting the right porosity, fiber alignment, stiffness, but crucially, they're trying to mimic the biochemical complexity too.
You mean including the right signals?
Precisely. These engineered matrices, or sometimes scaffolds made from decellularized natural tissues, where you take an organ, wash away all the cells, and are left with the natural ECM scaffold, need to provide the right cues. They need to incorporate the correct binding sites, like those RGD motifs we discussed, so cells can attach properly. They need to include components like specific proteoglycans that can bind and then release the necessary growth factors at the right time to guide cell behavior.
So you need the physical support and the instruction manual built in.
Absolutely. If you just put stem cells on a dumb scaffold, they might survive, but they won't necessarily organize into functional tissue. You need the smart scaffold, the ECM mimic, to provide the environmental cues that tell them how to differentiate correctly, how to align, how to connect with each other to form working tissue, not just a disorganized clump of cells or scar. The whole future of tissue engineering hinges on cracking the code of the ECM and rebuilding it effectively.
Incredible. So wrapping this all up, we started thinking of the ECM as just glue. Where have we ended up?
Well, we've moved miles beyond glue. We've seen it's this incredibly sophisticated, dynamically managed network of proteins and sugars. It's not passive background. It's arguably the chief regulator of cell behavior and overall tissue structure and function.
It determines strength through collagen, bounce back through elastin, and that crucial cushioning and hydration through proteoglycans.
Right, and it's the highway system for cell migration, the storage depot and switchboard for growth factor signaling, communicating constantly with cells via integrins.
And that fundamental point about healing really sticks out.
It does. The maintenance of normal tissue structure, the possibility of true regeneration versus just scarring, depends entirely on preserving the integrity of that underlying ECM scaffold, especially the basement membrane and stromal framework. If they're intact, regeneration is possible. If they're destroyed, the body can only patch things up with disorganized fibrosis.
So the next time you, the listener, hear about scarring or wound healing or even diseases like fibrosis or cancer spread, remember this incredibly complex and dynamic architecture that's orchestrating the whole process.
It's a constant battle and construction project at the microscopic level.
And if you had to pick one single piece that encapsulates the elegance of this system, maybe it's that RGD motif, that tiny 3-amino acid code arginine glycine aspartic acid.
It's certainly a powerful symbol of how molecular detail governs macroscopic structure and function.
The fact that our cells use that simple sequence via their integrin receptors to physically grab onto these huge matrix structures like fibronectin and laminin and translate that grip into internal signals, it really is the language connecting the cell to its world.
That microscopic handshake truly defines tissue form and function.
Which leads to a final provocative thought for you to consider. We know the ECM stores latent growth factors, and we know enzymes released during injury chew up the old matrix to activate them. So how much does the timing and the specificity of that ECM break down the careful, precise demolition of the old scaffold actually determine whether we get successful regeneration of new functional tissue or just end up with a scar?
That's a deep question. Is successful healing more about carefully building the new or about meticulously and correctly tearing down the old first?
Something to mull over. It suggests the demolition crew might be just as important as the construction crew in rebuilding our tissues.